Good morning, everyone. The fact that I'm wearing a tie in New York, but not from California. In Chicago instead. So even though we have the elite of the spinal cord world here in physicians and scientists, and maybe assumption that not everybody here spent their entire life studying spinal cord injury. So with your permission, I'm going to give you a little bit of history about spinal cord injury. This is, as a physician, I always have to give my disclosures. My disclosure for this talk is I was the PI of retreat studies of human transplantation. So I'm obviously very biased, and I'll admit that. The rest don't matter. So the first record of spinal cord injury was Imhotep in the Edwin Smith Papyrus. That was like 3,000 years ago, almost 5,000 years ago now. So he's credited with initiating the old world kingdom for Egyptologists in the crowd, and he built the Edifu castle. He was the architect of the Step Pyramid, the oldest monument known on Earth. He was elevated to a full deity in the 500 BC. We don't actually know if he was a practicing physician though. But when I say Imhotep, who do you think of? Yeah. So maybe one of the most brilliant people of that time is now really a bad guy. So Edwin Smith Papyrus actually has two mentions of spinal cord injury, and they're actually listed under the neck injury part of this. And this is what they're talking about. So this is the spinal cord injury, and this is the top of the spine. This is the bottom of the spine. They're supposed to line up. So here's the spinal canal, and this would be functionally a transection of the spinal cord. So this person would not be a candidate for any of our studies. Okay? So how did we treat that back then? Well, back then, if you initiated a treatment with, like, the Pharaoh, and you failed, they cut your hands off. So spinal cord injury, you did not treat. You died. Okay, so what happened in the next 5,000 years? This is a Civil War picture. What happened between Egypt and here? Not much. Not much. At that time, if you got a thoracic level injury, not cervical, thoracic, your life expectancy was 2 years. And you died from urinary tract infections, respiratory infections, and decubiti. Okay? So what really happened then was, in World War II and the Korean War, we developed good rehab, so you didn't get decubiti, good respiratory therapy, and we had antibiotics. So after that, your life for a thoracic level injury, even the low cervical, is now normal. If you're a C1 or a C2, you still only have about a 2-year life expectancy. Christopher Reeve lived a long time because he was very rich and very popular. So what happens, though, with the spinal cord injury? Even though we're living a full life now, you're still wheelchair bound, you've got restricted work, restricted sexuality. 50% of people with spinal cord injuries are not employed. So your life is very, very restricted. There are about 17,000 to 18,000 new spinal cord injuries just in the United States. There's about 250,000 to 500,000 people with spinal cord injuries living right now. The majority of them are young males, 80%, usually from falls or sports. Estimated cost per year is about $10 billion, and the estimated cost for the current individuals living with spinal cord injuries is $18 trillion, and that doesn't include the new injuries that are coming up. So who gets it? Well, interestingly enough, the majority of spinal cord injuries are young white males, and black males are the second most common is incomplete tetraplegia and incomplete paraplegia. So what happens to cost? If you're a 25-year-old male and you get a high cervical cord injury, your estimated cost for the rest of your life is $5 million. If we can change that to a incomplete, it goes down to about $1.5 million. So the impact of changing a high tetraplegic injury to a low is millions of dollars per individual. And multiply that times 18,000. And the importance of that is if you're a high cervical injury, if you're a C4 or above, you can't do anything for yourself. You need 100% assistance. If we can get you down to C7, you can do almost everything by yourself. So in cervical spine, if we can take the injury down 3 centimeters, we can dramatically impact your life. So this is what we do in research, right? So in device development, those of you who develop devices know there's three stages of acceptance of a device. The first stage is, it can't be done. Second stage is, well, he can do it, but nobody else can do that. And the third stage, yeah, that's my idea. All right, so where did this all start with? Well, I was lucky enough to finish my residency and go to the University of Florida, where Doug Anderson, Paul Ryer, and by the way, Ed Worth were at the time, and joined this phenomenal research group. And what they were demonstrating at that time was that they could take fetal tissue and completely reconstruct the spinal cord. So the tissues would grow and incorporate and demonstrate that you had improved motor function through numerous groups at that time. So that was very exciting work. But there's been a lot of other things. I'm going to talk to you about some of the stuff that's being done in the lab right now. It hasn't been translated to humans yet, because I think there's some very exciting stuff that is potentially translatable. So one of these is negative pressure. So for brain injury, they demonstrated that if they could create negative pressure, they could improve clinical outcome. And in animals, they demonstrated, let's go to the next slide. They demonstrated that they improved locomotor activity. And interestingly enough, the 50 millimeters of mercury was their best. So they had improved locomotor activity. They had improved fiber numbers. They had improved fiber length, fiber volume. And you can even see histologically with a typical injury here how much better the spinal cord looks. And that's just by creating negative pressure in the injured area of the injury. So that's kind of interesting. Well, how about oscillating electrical field stimulation? So this is an interesting group where they had three groups: normal, the spinal cord injury with an oscillating electrical field, and a spinal cord injury with just stimulation but no oscillation. And again, they did the typical things that we do, the BVB scores, inclined plate, all of that kind of stuff. And it was very interesting because you can follow this over time. So this is the spinal cord injury group without oscillation. This is with oscillation. And over time, the oscillating spinal cord stimulation group continued to get better in terms of the behavior and in terms of the inclined plate. And the motor evoked potentials continued to improve over time. And the myelin area also continued to improve. So that not just electrical stimulation, but an oscillating electrical stimulation. So imagine what we could do if we do a transplantation and cover that with an oscillating electrical field. Maybe we'll get some better results, right? Okay, how about transdermal electromagnetic stimulation? Again, same kind of thing. They demonstrated that by doing this, they could decrease the inflammatory markers in the macrophage. And by decreasing the inflammation, we should be able to improve our ultimate results because we're stopping secondary injury, right? So they did that. They implanted the stem cells with the seated scaffold, and they did the electrical stimulation. And what they demonstrated was, particularly right at the site of injury, they had a significant improvement in the distance that the transplanted cells would grow. Not much cephalad, but caudally, it continued to get better. And this is a very exciting one, I think, targeted activity-dependent spinal stimulation. So this is telling us how important rehabilitation is after any kind of spinal cord injury or a transplantation. So in this study, what they would do is they implanted electrodes, and if they could detect any kind of a stimulus going to the muscle, they put that to a program, which would then move that muscle automatically. So it received the impulse and it translated into a movement. And that they found was that over time, those animals improved 63% compared to the controls of 30%. And after they stopped the stimulation, that movement was retained. So this, again, hasn't been done yet in humans, but it's a very exciting possibility. And then there's another very new one called photobiomodulation, which seems to be going through the STAC3 pathway. And what they're demonstrating was they get improved at just with photostimulation. They get improved ambulation, they get less apoptosis, and they get greater cell survival. So that's a pretty non-invasive kind of thing, right? What about hypothermia? The football player a couple of years ago injured his spinal cord and was taken had hypothermia. We don't know if that actually had any effect on him or not, but he recovered. He was great, but we don't know. But it does. There is evidence that shows that it is effective. The behavioral scores are better, not at the site of injury, but proximal and distal to it. Cell survival is better. And there was a conversion rate from AGAs to a number of DCs and even Ds just with hypothermia. Okay, so what about the human studies? I'm just gonna, if I went through every human study that's been done, we'd be here all afternoon, and we'd all be asleep. So I'm gonna give you a summary of them in a minute, but I just wanted to highlight a few to kind of demonstrate what our problems are that we're seeing with the human studies. So in this one, by Kakabadze, we had autologous bone marrow aspirate from the iliac crest, spun down and harvested, 750 million mononuclear cells were injected, and they had 18 patients with thoracic and lumbar A, B, or C injuries. And they said, well, half of our patients improved 1 grade, and we had 2 patients improve 2 grades. So what are the problems with this particular study? Number one, it's a paid journal. So we have no idea what peer review meant for that. Number two, we expect 1 grade of improvement. So I'm not sure they actually showed anything. Number three, lumbar injuries are not spinal cord injuries, they're called equine injuries. And we expect them to recover. So the 2 patients who recovered to level, if they were caught equine, well, that's what we expect. So we have no way to tell who improved and who didn't in this kind of a study. This study was doing neural stem cells. It was phase 1 open with 4 patients that are chronic Asia A's. They did the typical parameters that we all follow. And what they demonstrated was it was a safe study. There were no major adverse events. And the one thing I'll say, in almost every study that's been done, it's been demonstrated that there have been no major adverse events, except for one. How about bioresorbable polymer scaffolds? So this is a porous bioresorbable scaffold. You seed that with your cells and transplant it. There were acute T11-12 fractures. Asia A's implanted at T12. No major adverse events. At 3 months, they improved from Asia from Asia A to an Asia C. Again, this isn't spinal cord injury. This is polyplant. So we don't know if that means anything or not. 6 months primary endpoint for the INSPIRE trial. It's a thoracic injury, approximately 15 patients. Bioresorbable polymer scaffolds, endpoint greater 25, and they had 50% conversion from Asia A to Asia B. That was a reasonably well done study, and it's encouraging for us to keep pursuing that. So this is, there's all kinds of ways to put these cells in. You can put it directly into parenchyma, you can put it into the CSF, you can do it IV, you can use mesenchymal stem cells, you can use neural stem cells, you can use oligodendrocytes, so there's all kinds. So I've broken these down so that you can see what is actually being done. So these are injections directly into the cord. 6 studies between 2008 and 2018. You can see there's a variety of acute and chronic injuries. It varies from cervical to thoracic to both, even with the scaffold. Relatively limited numbers of patients. Most of them are Asia A, one Asia A and B. This one we couldn't really tell. And their results were, either safe or maybe some slight improvements. Only one of these was done in the United States. Most of these are done in countries where we don't know what the quality of the paper is. How about intrathecal injection? The most famous one recently was the study done at Mayo with 1 patient, where the Asia A patient, and then they showed the patient walking. With 1 patient in intrathecal injection, we don't know what that really means. Maybe it's exciting. We just don't know. IV, couple studies demonstrated safety, and these studies had patients going from Asia A to Asia C. I would really like to see what's actually happening with the individual muscles, so that you can actually tell what's happening. To say you went from an Asia A to an Asia B, to me, really doesn't mean anything. It's not really a functional change. How about neural stem cells? There have been 3 studies in both cervical and thoracic, subacute and chronic primarily. Some change from Asia A's to Asia B's. These 2 studies were done in the United States. This study was done in Iran, and I don't know how to interpret that particular one. Schwann cells, all of these done in the United States. Thoracic levels, subacute and chronics, with a relatively limited number of patients, demonstrated primarily safety. Olfactory stem cells, or olfactory and sheathing cells rather, chronic injuries, thoracic and 1 cervical. This had 20, they showed no improvement. The problem with olfactory and sheathing cells is there is 1 published case of a creation of a tumor at the site of injury. I can tell you there have been many more than that. There's only 1 published. But boy, I tell you, that is ugly when you see that. So that's something we probably can't use. The OPCs, you're probably going to hear more about that today. I am biased. I think this study is the best and the most exciting study that I've seen in the literature, and obviously they're right. So what about the future? Well, to my friends, my clinician friends, my scientist friends, and to the investment community, what I can say is the future of my friends that get spinal cord injury is in your hands. Thank you.